We're going to change gears again. This time, I want you to imagine that we've designed PCR primers for a particular gene, and now we're going to measure that gene in a huge population. As we do this, we're going to see variants within the genomic sequence attributable to a particular gene that we're measuring. Some of these will be modifications of the sequence in intergenic space. Those might interfere with regulation of gene expression, but they don't alter the products themselves of gene expression. You might also have a sequence modification within the within a gene, but not in an exon, for example. Or you might have one at a wobble position within the coding sequence. In both cases, you have essentially no effect on the protein that would eventually be produced for that gene. But sometimes we see that we have a genetic variant within the exon in a position where having this letter, having this nucleotide, alters the amino acid that would be inserted into a protein at that location. We call these non-synonymous single nucleotide variations. Non-synonymous single nucleotide variations. It's much more compact to simply call it an nsSNV. So we want to know, for some novel sequence variant that we've detected through genetic screening, is it deleterious or is it tolerable? Is this gene causing an alteration to the protein's function that might be produced for this gene? So the story for how we pursue that answer dates back all the way to 2001. In a paper by Eng and Henikoff from 2001, the same people who put together the Blocks database and Blossom put together this database called SIFT that was intended to recognize whether a particular non-synonymous SNV was a probable or improbable substitution. So SIFT is a way of asking more or less whether this amino acid change that the non-synonymous SNV invokes, is that amino acid sequence change likely under the constraints of evolution? Okay. Now in 2002, the folks behind Polyphen, the Ramensky et al. Paper at the bottom, decided to add to our interpretation of these primary sequences by looking into the 3D structures that correspond to them. After all, we know that proteins don't function typically as just loose coils of sequence, but rather in a defined structure. So Polyphen, in 2002, was using our understanding of the 3D fold adopted by these sequences and the feature tables that had become part of the annotation for at least the human proteome and the yeast proteome and some of these major model organisms in trying to decide whether phenotype attributable to a particular structure was changing or not. And then finally, we see that SNAP in 2007 decided to add some rather aggressive new features in this space. Now, we already mentioned that 3D structure had been brought into the picture by Polyphen, but SNAP is able to do this in the context of solvent accessibility, which gives us the ability to include things like de novo sequence inference, a structure inference rather, for deciding how a sequence should fold up. Basically, solvent accessibility refers to the fact that when you have a globular protein, all the hydrophobic residues tend to be on the inside, and all the hydrophilic residues tend to be on the outside. This is kind of a partitioning that we see in how globular proteins operate. So we say that those on the outside, the part that's going to face water, are solvent accessible, and those on the inside are solvent inaccessible. So SNAP was going to use that kind of information to decide whether or not a non-synonymous SNV altered function. And it also brought machine learning. Machine learning is now entering into all kinds of bioinformatics disciplines. And we see that in 2007, people were already using machine learning as a way to calibrate the scores that came out of SNAP in order to understand whether we had a benign mutation or one that was going to completely destroy the function of that gene. So what are the kinds of things that we might ultimately use this amplicon sequencing process for? A lot of them have quantitation tied into them. So let us imagine that we have a tuberculosis patient who's produced an induced sputum, and we're trying to look at all of the MTB microbes that we can sequence out of those droplets to try to understand which of them are and which of them are not resistant to TB drugs. After all, it might be that multiple strains of MTB could be found in one person's lungs. And if you have a high fraction of these species that are resistant, that's a really bad bit of news for the prognosis. You need to be able to treat those, right? Similarly, you might imagine that a person that's host to multiple strains of MTB might start out with almost all of them being drug-susceptible strains. But as you treat them, especially if someone is very inconsistent in how they treat their TB, you might find that the fraction of drug-resistant strains is rising over the course of treatment. If so, you'd better change how that treatment is happening as early as you can to nuke TB from that person's lungs. So, how we quantify amplicon sequencing results may actually be a matter of life and death. Next up, we frequently see that sequencing is applied now for diagnosing the prognosis for cancer. After all, tumors typically are diverse in the genomic sequences that they have. We find that a cut from this side of the tumor and a cut from this side of the tumor may have different genomic rates of change that we see in them. So that is a big deal. Certainly being able to measure genes that we call drivers of cancer is something that we want to do to be able to evaluate the aggressiveness with which a cancer must be treated. And of course, there are plenty of ecological scenarios in which being able to evaluate the quantity of genomic variance matters. We've already talked about the case of operational taxonomic units. So being able to evaluate whether the different pools that have different pH values show changes in what their microbial diversities, that we see differences in beta diversity as a function of pH. Being able to answer that as a quantitative question is a matter of some importance. So let's spend a brief moment to talk about calibration curves, because this shows up all over the place. Now, in this case, we have a measure of reality, which here is shown on the x-axis, the DNA concentration. So we know that at the left end of this curve, we have a very low DNA concentration, and we know at the right, we have a very high DNA concentration. Frequently, this is shown on log scale to reflect a really large dynamic range for this particular kind of measurement. So we might have, for example, 3 times as much DNA as at each push to the right as we go across DNA concentration. And then for that, we have some kind of detection. We've talked about fluorescence detections, but in this case, we're counting how many reads did we have for this particular bit of DNA in response to a rise in its concentration against some background that's relatively unchanging. Over at the left of the scale, we have so little DNA there that we may not be able to detect it at all. It may be that we end up with 0 reads for the DNA. That does not mean there's 0 copies of the DNA present in the sample. It just means that from our sequencing depth, we're not able to see it. So that means that we've fallen below the level of detection, the LOD, for measuring the DNA concentration by read count. On the other hand, you may have a period where the read count kind of bounces up and down in the grass before it starts its steady climb upward. This may reflect that we're below the level of quantitation. Generally speaking, in any quantitative technology, we feel most comfortable about our measurements in this range of linearity, which is to say that with log fold changes in the amount of DNA concentration, we get log fold changes in the number of reads produced for that DNA. So whenever we're dealing with some sort of quantitative technology, being able to interpret the reads or to interpret the fluorescence intensity or to deal with chromatographic intensity for that matter, we want to see a range of linearity where log fold changes in the underlying concentration lead to log fold changes in our measurements from it. So why else might we see that counts are misleading us in these? We see that different target sequences may have different primer efficiencies. So if you're trying to measure multiple DNA targets, you're amplifying different target sequences, you may find that you have different primer efficiencies for different regions, and therefore the concentration of one is rising linearly and the other is not. Well, that would be pretty bad. So having different efficiencies makes it very difficult to compare between different products. It might also be the case, rather, that sequencing errors would cause us to have to think that some particular variant existed even when it doesn't. Remember that we saw that the final algorithm we looked at for OTUs was screening out the singletons. One of the reasons behind this is that you might misinterpret a sequence that you saw only once as some genetic variant that you thought was very, very interesting. So that's problematic. And finally, it might be that stochastic noise might cause you to believe cohorts are different when they are not. Well, that's a big term basically for saying random fluctuations. So you may simply run, if you were to run the same sample multiple times, you might see differences in the alpha diversity within each of those, even though they represented sequencing from exactly the same sample. So it may be that random events, such as which templates get chosen for sequencing, may cause you to have what appears to be a significant variation even when it's not. So we must be careful with the statistics we use for evaluating whether or not differences exist between these populations. So there are a lot of takeaway messages from this lecture. And I just want to point out that bioinformatic tools exist to support you in a lot of places as you're doing amplicon sequencing. The process of selecting PCR primers, we looked at Oligo7 and the tools that are used to back the NCBI website as well. Certainly controlling the quality of sequencing is a very big deal. Fred and FastQC have been around for a long time now to help us to understand how quality scores vary as we get to longer and longer sequences. Being able to trim away error-prone flanking sequences is one of the outcomes from that. You would try to use the Fred scores to guide you in how to trim the DNA sequencing reads. Being able to cluster the reads to determine which are the distinct sets of sequences, the OTUs, that can be derived from it, is certainly a major application of bioinformatics. We talked about 3 different tool sets that have been created for that purpose. Being able to match those clusters to taxonomies, of course, matters. Being able to interpret the data requires that kind of processing. And being able to determine the completeness of sequencing through rarefaction analysis is valuable to you. So being able to subsample your data to determine how much diversity changes in response is appropriate. Evaluating the disruption of function by mutation is a highly significant process. Polyphen and SIFT and all of the tools that have been created in that family are a very big deal and can always use more insights as to how to make them work ever more effectively in particular contexts, such as understanding which mutations are most likely to render a particular microbe to be resistant to the possible treatments we have for infection. And finally, being able to quantify particular sequence variants is something that we've already begun developing good statistical models for as well. So I hope that these little packages of lecture have been really useful to you in understanding all the different tools that exist for amplicon sequencing, and I hope that you find that your research is greatly improved by knowing about them. Thank you very much.